Please be seated. Good afternoon. Is it evening? My apologies. Almost evening. And I presume, Professor Herselman, you have organized stuff for evening.
I did.
Thank you. Then it is evening. Dear friends, colleagues, distinguished guests, good afternoon, good evening. Welcome to the University of the Free State. It is absolutely fantastic. We are almost full. Last week we were half this number. Today, Professor Herselman, we are about to fill this hall, and it's wonderful to see this because we haven't had it for over 2 years. May I be allowed to make a special welcome to the extended family and friends of Professor Liezel Herselman. Particularly want to recognize in our midst Mr. Ben Herselman. I don't know where you are, sir. Welcome. And Mrs. Hanneke Herselman, the mother of our distinguished professor this evening. Welcome to you both and to the other smaller, younger professor in the making. In addition, tonight, this evening, we have our dean, Professor Dani Vermeulen. We have our distinguished speaker this evening. We have a whole range of colleagues from the faculty, from our university, and beyond. It really is a fantastic occasion, and thank you for making it. Colleagues and friends, an inaugural lecture is an auspicious occasion for the university and for the individual who's delivering it. It acknowledges and celebrates, and in particular welcomes the appointment or promotion of a new individual to the rank of full professor within the academy. It also represents a significant milestone in an academic's career. That is why we should be very careful, in my humble opinion, in the usage of the term professor, because those that have earned it, and those that have yet to earn it need to respect it. It provides the individual an opportunity for them to inform and to update the academic community on their current and future research, their life's work, and what they will intend to do in the future. The lecture also provides an added benefit to us as a university, to the individual presenting, to share the work that we do with broader audiences, thus helping us to improve the wider awareness of the university's impact on society. We do research not just merely for the sake of advancing knowledge, but we do research to transform people's lives. And this you will see, ladies and gentlemen, later on this evening. The Faculty of Natural and Agricultural Sciences at our university currently has 37 full professors. Professor Herselman, if I am not mistaken, it is the highest within our university. Today we welcome and introduce Professor Liezel Herselman through this public ceremony and the delivery of her inaugural address with that very intriguing title, The Ongoing Titanic Battle. I think she just forgot the word Titanic. Against destructive serial diseases. This battle ongoing between pathogen and host. And I'm sure she is on the right side of the host. Colleagues, ladies and gentlemen, it is my singular privilege to request Professor Dani Vermeulen, our Dean of the Faculty of Natural and Agricultural Sciences, to introduce Professor Liezel Herselman. Thank you.
Dear Dr. Malapu, thank you very much for the privilege that I have to introduce Professor Herselman. I was given 5 minutes to speak about her, but the people in the faculty will know that's impossible. It will take a little bit longer than 5 minutes to introduce her, but we'll try to stick to the time as much as possible. It is really a great privilege to introduce Professor Herselman to you, especially because we worked so closely together the last 7-8 years. But before I start, I just want to say to her parents and the sisters, there's such a lot to say about a distinguished lady like Liezel, and you can be very, very proud of her. And I'm so glad that you are all here tonight, and we are really honored to have you here tonight. Now, it's always difficult to speak after Dr. Malapu. He's got such a nice accent, and my Boere accent uit die Karoo is very different from his. So, but please bear with me, because I mean, we always joke and say English is the enemy's language. It's not really true, but we that grew up in Afrikaans, and sometimes we speak a black language even better than English. So just bear with me tonight, and I hope you will all enjoy the evening, and we're really looking forward to Liezel's talk. Now, when I asked Liezel for a short CV so that I can introduce her, there was a few things about her that even I didn't know. So I'm going to go through all this, and I will start with the bad one, is that she spent her first 34 years in Potchefstroom. Now, that's not a great start to life. I mean, if she was in Bloemfontein or wherever, well, you could have done worse in Pretoria. So I'm glad for that one. Then she attended the Bailey Park Primary School and the Potchefstroom Gymnasium Secondary School, and was a school prefect in matric. And all my HODs will know that's not a surprise, that she was a prefect. Now, then she obtained a BSc degree cum laude, but that's also not a surprise, in 1992 from the then Potchefstroom University of Christian Higher Education, now the University of the Northwest. She also received a Faculty's Award for the Best BSc Graduandus in 1992. She went on to obtain a MSc degree in biochemistry. I thought she was a plant scientist, and then I came up with this biochemistry. So of course, there are places where we can use her, right? With specialization in biotechnology, also cum laude, in 1995, also from the University of Northwest. And then she obtained a PhD degree from the University of Greenwich in the UK. With the very long title, I first thought I would read the title, but it's such a long, difficult title, I decided she can mention the title herself when she has a speech. So then she started a career in 1994 as an assistant agricultural researcher at the ARC, which is a very good foundation for any scientist. That was in Potchefstroom as well. She stayed in Potchefstroom and then gained experience on working on different crop species, for example, dry beans, lupins, sunflower, groundnut, and she obtained experience on working with different molecular techniques applied in plant breeding. In 2004, Liezel was appointed a senior lecturer here at Bloemfontein at the university, and she was promoted to an associate professor in 2008, and she became a professor in 2021, last year. Since 2013, she served as the academic departmental head of plant sciences, and this is where we started to cross our paths, and where she became, and I had to go and look up this word because I'm not sure if it's the correct word, so unmissable to the department, unmissbar. That's the word I was looking up for the whole faculty, to be unmissable for our faculty, and I think everybody will agree on that one. Her research career over the past years mainly focused on the application of molecular biology techniques in different breeding programs. Her current research mainly focuses on 3 areas, namely genetic fingerprinting of crops, important to the African continent, genetic analysis of plant pathogens, and marker-assisted resistance breeding. Something that I don't understand where I come from, but I can read it. Another part of her research work focuses on molecular marker techniques applied to plant fungi and bacteria, for example, genetic fingerprinting of South African stem and leaf rust pathotypes using molecular markers, everything around molecular markers. However, the main focus of her research is marker-assisted resistance breeding within a South African context. Currently, her research projects focus on resistance breeding in wheat. The main objective of the project she works on is the development of South African cultivars with durable rust and FHB resistance, adapted to South African conditions. Focus is also placed on bread-making qualities of these developed lines. I must admit that she gave me a lot more information, but I did not understand most of it, and some of the terms I will not be able to pronounce. So rather than making a fool of myself, I will rather let her do the explaining later. Can we agree on that one? Thank you. Okay. On her scientific outputs, she's got a Scopus, an H-index of 20. For people that's in academic world, they will know what it means. She graduated 13 MSc and 21 PhD students as a supervisor and co-supervisor. She published 50 articles in international peer-reviewed journals with 83 contributions at national and international conferences. Quite an achievement. But the greatest award was in 2020, the South African Plant Breeders Association bestowed a prestigious Fellow Award on her in recognition of her exceptional contributions to plant breeding in Southern Africa, especially training and mentoring the plant breeders of the future. A true academic. Now we come to the fun facts. And she gave me a few, but I've got a few more. So the first one she told me, a fun fact, is she loves her family. And I'm pretty sure they love her as well. Huh? That's the first one. The second one is she completed 2 Comrade marathons. She completed 2 Argus cycle tours. And she did 1 mid-mile. Obviously, she didn't drown, otherwise she wouldn't have been here. Huh? So, she enjoyed reading, and I asked her this morning when she was in my office, what is the real English word for speurverhalen? In Afrikaans, we know it's detective stories, but it doesn't sound right. Speurverhalen, that's what she loves doing, and building puzzles. I don't know where that one comes from, Liezel, but yeah. And she loves working in her garden. I know how beautiful her garden is. She loves to go out for a red cappuccino and or a meal. But she doesn't like whiskey, and I will come to that one later. She's a dog lover. That one we all know. She's got 2 dogs, Kuru and Grace, and I had to ask her this morning, what does Kuru means? And she told me in Japanese it's black, but of course the dog is black. Now, between herself and our faculty teaching and learning manager, Elsmarie, we all know everything about raising dogs and looking after each other's dogs. After every meeting in a faculty, they spend hours talking about their dogs and exchanging photographs. And we all have to be part of that. Now,
you do that with your grandchildren.
I'm allowed to do that, but Prof Hendrik beats me on that one. Eh? And then there's one more thing that I need to talk about. There's a very distinguished group, small group of academics, the Qpcr group. That's the HODs that's got to go to Qpcr on a regular basis, and we always go in a little bus. And we've got a motto that what happens in that bus stays in that bus. But there are a few things that I maybe should mention tonight. Now, the first one regarding the whiskey. Every time we go there, one person must buy the bottle of whiskey. And even Liezel, who doesn't drink whiskey, she doesn't drink, had to buy a bottle of whiskey. And she's still blushing today for having to go into off sales to buy that bottle of whiskey. That was one of the hardest things, and she still complains about the price of the whiskey. But she had to do it then. And then there's one more, and that's the one that very few people know. She loves to eat the Smarties color by color. Now, some of the people told me I'm exactly the same, but I have to look here somewhere in my pocket, and I had to buy a small box of Smarties. So tonight, or when the family's gone to bed and she doesn't want to build a puzzle, she can count and sort the Smarties in the box. So that is from the Qpcr group, Liezel, your box of Smarties. So, okay, now we come to the serious part, and I'm nearly done. There's a very good reason why we affectionately call Liezel the hoofdochter of the faculty, the head girl. She's always well prepared. She reads through all the minutes of meetings in detail. She's always prepared. I mean, I can just ask, Liezel, is the minutes correct? And she will say yes, because she read it in detail. And she always provide brilliant inputs in our meetings. Apart from being the academic departmental head of plant sciences, she serves on the executive committee of the faculty. She serves on the committee for revision of the promotion criteria of the UFS. And since the beginning of this year, she also serves on the executive council of Senate, which is quite a great honor. So she's really a great academic. A part of being a scientist of note, she's an excellent leader. I actually wrote here a fantastic leader. As you all know, I love to look for applicable quotes on people, and I always try to find quotes from famous people, but I battled with this one. The other night I tried to find out something about quotes regarding a stalwart. Afrikaans, a staatmaker. A staatmaker sounds nicer than a stalwart. But I couldn't find any quotes regarding a stalwart. But I could find a few synonyms to what a stalwart means. And I'm going to go through them. The first one says dependable. That's Liezel. Huh? The second one, fearless. That's also Liezel. The third was, and I had to go and look up these English words because my English aren't that good. It said indomitable, ontembaar. But that's not Liezel. No, that one doesn't sound right. Interpret, onverschrokken. That's Liezel. Staunch, standvastig. That's also Liezel. Stout, in Afrikaans it's been written like stout, which means strong of character, brave, firm, and obstinate. That's also Liezel. And then finally, tenacious, which in Afrikaans is taai and vasbyt. Running 2 marathon comrades, doing 2 cycle tours, swimming the midmar, that's vasbyt. And that's Liezel. Then I also, that same night, start to look at quotes for leadership. Regarding leadership, we all in the faculty value her skills as a leader. And I could find a lot of applicable leadership quotes. But there's one guy, and I've read a lot of his quotes in the past from John Maxwell. He does a lot of quotes on leadership. And his summary regarding leadership was, being a great leader is all about having a genuine willingness and a true commitment to lead others to achieve a common vision and goals through positive influence. No leader can ever achieve anything great or long-lasting all alone. Teamwork goes hand in hand with leadership. Leadership is about people and for the people. And he ends that quote with, a true leader is one who knows the way, goes the way, and shows the way. And then I found another quote from him. Let me be that the final one. He says, the pessimist complains about the wind. The optimist expects the wind to change. But the true leader adjusts the sails. And that's Liezel. She always adjusts the sails. This is how I can just describe this exceptional person. A great friend and a true colleague. Liezel, we're looking forward to tonight. Congratulations with your promotion and all your achievements. We will give you as much time as you want tonight, not just the 40 minutes. We're very worried about the 40 minutes. We will give you enough time. You can talk as long as you want. And good luck, and we're looking forward to your lecture. Thank you.
Good evening, everyone, and I want to thank everyone for being here and sharing this special day with me. And as already said, I'm very grateful that my parents can still be here and share this with me as well, and the rest of my family. And also a word of welcome of those colleagues and friends attending online via the live streaming. So thank you and welcome to everyone. I also want to say a special word of welcome to Prof Dani. He's celebrating his birthday today, so congratulations, Prof Dani. And also, I heard that Prof Koos Albertijn is also your birthday today, so congratulations. I wanted it to be a little bit darker for the next slide, but because of all the lightning, just bear with me. It's not totally what I wanted, but here we go. So my title is, as mentioned, is The Ongoing Battle Against Destructive Serial Killers. Yes, we have serial killers in the room. So we have to deal with 4 destructive serial killers today. The serial is wheat, and the killers are 4 fungal diseases. So in this time, and as I said, it's a battle against the serial diseases. So as time is ticking, we can ask the question as to, I just want to see if the mouse is working, yes. We can ask the question as to whether the fungal disease is winning this race, or is it the wheat plant, or is it us as scientists, the conventional breeders, the plant pathologists, or the molecular plant breeders. Now, hopefully at the end of my lecture, you will be able to answer this question as to who's winning this battle. So, because we have a lot of postgraduate students also attending today, I want to inspire them with the path that took me to where I am today. And I want to motivate them and say that often where you start in your career is not where you end. But being at the university lies the foundation of and gives you all the necessary knowledge and experience to be successful in your career. So I want to start with where I start with my career. So I started with my BSc, majoring in biochemistry and botany. And just for fun, I thought, let's do some chemistry as well. And you can see no plant breeding in there, but the 3 subjects together gave me a good foundation to become a molecular plant breeder. So from there, I continued on with my master's degree in biochemistry with specialization in biotechnology. And I worked on a disease, but a human disease. And this disease is caused by a single base pair change in the human genome. And it's known as Osteogenesis Imperfecta, or brittle bone disease. And this disease causes children from a very young age, even from birth, to have broken bones. And just a fun fact is that the actor from the series The Middle, Atticus Schaffer, is also being diagnosed with Osteogenesis Imperfecta. From there, I started working at the Agricultural Research Council, and I started working on groundnuts or peanuts, once again in a disease, but this time it was a viral disease. And this viral disease is transmitted by aphids, and it causes groundnut rosette disease. So in my PhD, I tried to find markers linked to the resistance genes in peanuts. Then, when I started working at the university, my focus shifted to wheat, because wheat is being produced in the Free State, the Northern Cape, and I started working on that, and then specifically on some of the fungal diseases that causes a lot of destruction on wheat. So that's how my focus shifted. And so tonight I will speak about these 4 serial killers or diseases: the Fusarium head blight and the 3 rusts: stem rust, wheat rust, and stripe rust. So before I go to that, I just want to give you an idea of some of the past projects that shaped my career and gave me the knowledge and the information to do what I'm doing today. Many of those I've done as a main principal investigator, but some of them was because I was exposed to some of the projects my students worked on. So the first crop I worked on was common bean. We all eat a nice boinkie sop in winter. Then I moved on did some work on sunflower, and then of course that followed by peanuts, on which I did my PhD. Then when I started working at the university, One of my first projects was on coffee, quite an interesting crop to work on, and I was also exposed to the new world of cassava and also mangoes. With mangoes, I had an opportunity to travel to Mozambique. And then, of course, what will breeding be without working on maize? I also had opportunity to work on cotton and sorghum. And then, of course, knaf. Now it's not wheat that you can smoke, it's just knaf. I did some work on cactus pear, then also on cocoyam, clivia, the beautiful clivia, and some work on butternuts. So all those shaped my career for being what I'm doing today. So the question is, why plant breeding? And here I would like to take a page of my Dean's book where he loves to quote people. So I looked for some quotes on plant breeding, and I found this quote by Luther Burbank, who was an American botanist, horticulturist, and a pioneer in agricultural sciences. And through his career of 55 years, he developed more than 800 variants and strains in different plant species. And he stated that the secret of improved plant breeding, apart from scientific knowledge, is love. And I can tell you, I love being a molecular plant breeder. And then just to throw in a little bit of a fun fact, something funny, where normal people like to pop bubble wrap, plant breeders emasculate flowers to do crosses. Then I would like to have 3 more quotes. I can't have just one quote, Prof. Dani. And I'm just going to move around because I didn't learn this out of my head. So it was from the former US Secretary of State, John Kerry, and he stated that food drives and life. I can't see there, sorry, even if I have my glasses on. Food drives life, and the struggle for the food is a struggle for life. Can you still hear me? Okay, thank you. This makes hunger an economic issue, a national security issue, and without a doubt, a moral issue. Through innovation, we can help elevate hunger and malnutrition today, but more than that, we can help fulfill our responsibility to tomorrow. Then the Food and Agriculture Organization of the United Nations says that plant breeding is one of the shortest and most effective routes to improving global health through better nutrition. And then my last quote from Bill Gates, and I'm sure you all know who Bill Gates is. He said that I've seen firsthand that agricultural science has enormous potential to increase the yields of small farmers and lift them out of hunger and poverty. So that brings us to my problem statement of my research. So I work on wheat, and debatably we can say that wheat is one of the most important food crops, and also because it produces a lot of carbohydrates. But unfortunately, the production of wheat is then challenged or threatened by the cereal killers that I'm going to address today, and specifically the 3 rusts, the stem, stirrup, and leaf rust, as well as Fusarium head blight. And these cereal killers is putting food security at risk, but also that the livelihood of the producers. And the world population is estimated on an average is growing about 81 million people per year. And the arable land available for production is not increasing, and in some instances it's decreasing because of urbanization. So one of the aims of my research is to produce plants that's got resistance against diseases that will increase yield on the current available land that we have available to do breeding on. So now the question is, how do we stop these cereal killers? The best way to stop a killer is to stop before it can even start. So we do that by incorporating a number of resistance genes into that plant and providing it with mechanisms that can protect the plant. It's very similar to protecting your house against thieves, where you have some locks, you have a security gate, and you have an alarm system. Not that it always works, but the same for the plants. We might introduce some genes, but it's not always helpful because the pathogen can overcome all the barriers that we've put there for him to prevent infecting the plant. So we are For a molecular plant breeder, Prof. Dani, as you said, it's all in the genes. Because the gene will then tell us whether we have resistance or not. And this is based on Flor's gene for gene hypothesis. And for the chemists and the physicists here, this is very similar to Newton's third law, that for every action, there is a similar reaction. And that's the same. For every gene present within the plant that provides resistance in the plant, there's also a gene in the pathogen that will not be able, the pathogen then can either be resistant or susceptible. If the plant has got a gene that the pathogen can't overcome, then the pathogen won't have any effect on the plant, and then vice versa. So that's what we base all of this work on. And so we focus also on genes that's present in other wheat cultivars and closely related grass species, so no GMOs, just conventional breeding with available genes that we have. And we also want to try and incorporate a multiple number of defense mechanisms into the plants to make sure that they can really protect themselves against the diseases. And then we focus on genes that provide resistance at an early stage in the plant, so at seedling stage throughout the plant's life, but also some genes are only effective at adult plant stage. So we try to focus on all of these different factors. If we look at wheat production in South Africa, is that wheat is the second most important crop in South Africa, but production of wheat is declining for a number of reasons in South Africa, but also in the Free State. It's because of the high input costs, and the war in Ukraine is not helping this. Also because of the low commodity prices, and then also the diseases that causes the farmers very difficult for them to cultivate wheat. So because of all of that, South Africa is a net importer of wheat, and that's making it a little bit difficult. So we are focusing on diseases that will help the farmers to overcome some of the problems that they experience in producing their wheat species. So if we first look at the rust, now very similar to COVID-19, where we have different variants, started at the Alpha, then the Delta, that was very serious. Now we're already at Omicron. It changes, not all of them are as serious as others, and we have the same in the wheat rust. We keep on getting newer and newer races. They change over time. Some of them are more serious for the plant, some are less. So we have to continuously have a battle against all these new races that develop to make sure that we have plants that's resistant against these diseases, and that we can still have yield and have some flour at the end to bake bread. So how do we, most of the times we have a single gene that provides resistance against a specific race. But that is not very effective, as you've seen with Omicron and the COVID-19, it overcomes the resistance. So the focus are now shifting to more durable resistance, where we make use of a combination of genes, or where we use genes that's only effective at adult plant stage and that only get partial resistance, but that's much more difficult to overcome and gives the plant a much higher chance of surviving this disease. So that's the focus for the rust resistance. Now, the fourth serial killer is the Fusarium head blight, and that's a very difficult one to control because there's no registered fungicides for this disease in South Africa yet. It's a disease that's all over the place. It's dependent on weather conditions, and you can know the weather is not predictable, so the disease is not predictable, and also there's also a wide host range. That Fusarium also occurs on maize, and the farmers do intercropping between wheat and maize, and that causes the fungal to be debris to build up in the farmer's field, so it's very difficult then to control Fusarium head blight. But luckily, with breeding, we are able to incorporate resistance genes into the plants to protect them, and that's more promising, more cost-effective, and we also then can reduce the epidemics there. So then, finally, we then get to the aim of my current research, and that is to stack or combine different rust and FHB resistance genes into locally adapted South African cultivars, so that we can go from a diseased plant like that to one that's showing no disease or that show good tolerance towards the disease. So throughout my breeding program, my focus has been on the wheat rusts and the 3 different wheat rusts. Now, I listed a number of wheat genes here. You don't have to know all of them, but what's important that for the leaf rust, we focused on genes that's either giving resistance from seeding stage throughout the plant's life, and we also looked at the more durable adult plant resistance genes, and that's the same for the stem rust and also for the stripe rust. For the stem rust, we also had to look at genes that's effective against one of the newest variants of stem rust, UG99. We had a long time no problems with stem rust, and then around 1999-2000, a new variant was discovered in Kenya and Uganda, what was first typed, and Prof Saki Pretorius, a former head of our department, played a big role in describing the new race. So we had to look at some genes to incorporate there that will give resistance against this new race. And for the stripe rust, we focused on some genes present in the South African cultivar Kariga that's been planted widely and providing some nice extra resistance there. For the Fusarium head blight, our focus is on 2 types of resistance. The plant can show resistance against initial infection that prevents the infection at all happening, and that there we are looking at 2 different genes there, but there's also type 2 resistance, that's a resistance against the spread of the disease once it's infected, and there we are looking at incorporating 3 different genes in our wheat plants. So the focus of the pre-breeding program is to apply markers that are linked to these traits. So with marker-assisted breeding, it's all based on there are certain genes that will give resistance, but we can't always detect the genes directly. So we have some markers in our genome that are linked to that gene, and we can follow that in our breeding program, and then we can see, yes, we've incorporated the genes or not. And because we are combining a number of resistance genes, it's much easier if we have markers, because we won't be able to phenotypically always see whether the plant has 3 or 4 genes present. So we have all of these techniques that we can use to do that. So we started the pre-breeding program to combine the different resistance genes, and then for the different diseases, the leaf rust and also the Fusarium head blight. And we try to then incorporate these, make these lines available to breeding programs, but also to confirm these resistance in the greenhouse and in the field. We can't just rely on our marker data in the lab. So the question is then also, where did this all start? And this started with a paper in the drawer of Prof Saki, and we started to discuss the possibility of combining different rust-resistant genes. So it started where we had 4 different parents that we identified, and each of those parents contained a different number of the rust-resistant genes. So in the first cross, we crossed 2 of these parents, and we got the offspring, and then we selected those with the most number of markers based on the marker data. We did the same for our second cross, where we also developed lines. We selected the best lines, and then the 2 best lines were then crossed with each other to develop a double-cross population that we then self-pollinated to get the genes in a more homozygous state. Throughout this whole process, we made use of markers to follow the integration of these genes. So most of our breeding schemes that we use are based on this principle. So when we did that, we were able to identify the 2 best families that contained the most number of genes present. And you can see that some of the genes that were absent in family 1 was present in family 2, and then vice versa as well. And then we knew that if we can combine these 2 families, we cross them, then we have a chance of getting all those genes into one cultivar, and that was our main aim there. After we've done that, we had to test and see if the gene was really transferred, and if we see the gene, is it really present. So we did some phenotypic screening with the help of our pathologists, and you can see that in family 1, it's very small on my screen here. For the first is the stripe rust. So family 1, for the markers we knew contained a stripe rust resistant gene, and you can see family 2 showed no infection, and that gene originated from this parental line. So the phenotypic data confirmed our marker data. And the same happened for the leaf rust, where both the families contain 2 leaf rust resistant genes, and you can see both of them showed nice resistance, and then this was the line that was used to transfer the resistance gene. When we came to the stem rust, we expected the family 2 to contain the gene, but it was not present, and then we realized that our source blade was not totally homozygous, and that caused some problems, that sometimes it was transferred and sometimes not. But this was the first step, and we introduced a number of rust-resistant genes into our wheat lines. Then came UG99, and we also had to be proactive and try to combat this new variant. So we looked at introducing resistance. We screened different parents. You can see Dunphy here and Kingbird, and obviously Kingbird showed much better resistance against the disease. So we decided to incorporate Kingbird in our breeding program. So we've identified family 1 and 2. You can see there's family 1 and 2 with the different rust resistance genes. And then family 1, we crossed with a line that contained SR39 that provides resistance against this UG99 variant. And the other family we crossed with Kingbird. Once again, we followed the same breeding scheme, and with markers at the end, we were able to select lines that contained the original rust-resistant genes, but also incorporated that SR39 and the resistance from Kingbird. But if this was not enough, we decided, let's try and also introduce Fusarium head blight resistance into these lines. And here you can see the example of, in this slide, after the plant was infected in the spike, you can see the progress of the disease, and you end up with a spikelet that's totally infected. Compared to a plant that's showing some sort of tolerance, the infection didn't really spread from where it was introduced with the disease. So this is what we want to see after we've done our breeding. So once again, we used our 2 rust families with the different genes, and this time we crossed it in one example with a line that contained type 2 resistance, and then we also crossed it with Frontana that contains type 1 resistance, so that we can combine as many as possible resistance into these wheat lines. Followed the same process, and at the end we could select lines that contain both rust and Fusarium head blight resistance in our breeding program. But simultaneously to this, what we also did is we know that Crocodile at that stage was a cultivar that was giving good yields and that was favorable to plant. So we said, let's try and incorporate Fusarium head blight resistance into this Crocodile line. So we crossed the Crocodile with the donor of the Fusarium head blight, and then we kept on back crossing to Crocodile to ensure that if we get to about a backcross population of 6 generations, we will have Crocodile that's very similar to the original Crocodile we started with, but now containing the resistance gene. Because this resistance source is from overseas, it's not adapted to South African conditions, so we want it to be very similar to Crocodile, but with the resistant gene. So that is what we did here, and throughout this whole process, we followed this again with markers to make sure that we select for the resistance gene, but that we also select for the genetic makeup of Crocodile. Then we start, now we have a nice line, South African line with Crocodile in its background with resistance. Let's try and then add and combine the Fusarium head blight resistance in these lines with the rust resistance that we've already developed. So to do that, we use these lines that we developed with the Crocodile background. We looked at those lines and we selected 26 lines that showed the best resistance and the highest number of resistant genes. And we screened those with the F6 FHB markers, but also we said that we can't just breed a line that's got resistance, but does not have good bread making quality, because at the end of the day, we need to bake bread with the lines. So we also looked at markers that's linked to good quality traits. So we selected, based on that, then 2 lines that showed good Fusarium head blight resistance, but also good baking quality traits. And then these lines we then use further in our breeding program. So next up, what we wanted to do, we took our best rust-resistant lines and we crossed that with this Fusarium head blight resistant lines that contain those resistance. And then we followed a little bit of a more integrated or a type of breeding approach where we crossed those two lines. We got offspring. We either crossed that with themselves or we selved them to get them in a homozygous state, or we used the progeny here and we crossed that back again to crocodile, or also we selved that, or we used the two double crosses to cross. It's just different possibilities with different end results in getting the combination of our genes that we want. We also introduced through one of the lecturers, who was then one of my students, through Ansari, we introduced the double haploid technique where we would get the lines in a more homozygous state that's more preferable if we want to continue with the breeding lines. And then at the end, we developed different populations that we either could backcross to do the to make it more homozygous, or use it in other backcrosses. So this is still an ongoing process because we keep on identifying the best lines and the lines to continue with. But simultaneously, we wanted to improve the FHB resistance as well. So we identified some of the lines with the best FHB resistance, and we again crossed that to reduce double crosses, crossed it back to crocodile, or crossed it with each other to develop lines that we then also could use further in different breeding schemes. So the current and the future breeding scheme that we are using, then we have identified 3 rust lines that contains most of the resistant genes, and we identified 2 of the Fusarium head blight lines that contain a nice resistance against FHB. We are trying to develop double haploid lines, follow the integration with markers, then look at baking quality with the use of some other biochemical techniques, and also follow the presence of the disease with Qpcr. And then best lines being identified, then again crossed with each other, and the incorporation of the genes being followed. So this is also an ongoing process of one of our current projects. So throughout this whole breeding scheme and all the different breeding that we've done, we've used a number of techniques that we need to be able to be successful at the end of the day. So the first is our phenotypic screening, once again with the help of our pathologist, where we can identify the lines that show resistance and see if they are really, that correlates with our marker data. Then we also did the marker-assisted selection, where we used the markers to follow the number of genes transferred. And this is one of the double-cross populations that we developed. And you can see the most lines contained either 3 or 4 of the markers, but we did find a few lines that contained 8 of these markers. And then in our more recent populations, we saw the same trend. Most of the lines contained 3 or 4 genes, but we did identify some of the lines that contained 8 or 9 of the genes. Unfortunately, none that had 10. But these lines we now can identify, or we can work with them. So this is just, you don't need to know all the numbers here, but just to indicate that some of the lines contained 9 genes, some of them contained 8. They have different combinations, and based on our marker data, now we can select the best lines to cross with each other to make sure that we now combine all those resistance genes into a single plant. Then one of the techniques that we also, with the help of Ansari and Botma, started doing is our Qpcr, and I think most of us would these days understand or know Qpcr, because if we want to know if we COVID negative or positive, we go for a Qpcr test. And that is the same that we are doing here. We want to see if the plant has the fungal disease in it or not. So we use Qpcr for the same process. So what we did is we phenotyped the lines using some of the most relevant rust races that we currently have, and also the Fusarium head blight. We use both the spike and the spray inoculation to test for type 1 and type 2 resistance. Then we did our Qpcr on these lines, and we used the beta-tubulin gene of the pathogen to see if it's there, and we had to have a reference gene in our wheat plant to compare it with. And then these are the lines that we tested, and these are the marker data that we had in the lab. So we wanted to see, Do we see a correlation between our marker data, the Qpcr data, and also the phenotypic evaluation? So I'm only going to show you the results of leaf rust because we saw the same trends. I don't need to show you all the different rusts. So with leaf rust, we did the phenotypic screening, and we could see that the CS line that contains a leaf rust showed no infection. When we compared that with our marker data, we could see that this line contained 2 leaf rust resistant genes. And when we did our Qpcr, we saw that the best results are obtained 10 days post-inoculation. And you can see this line is also one of the lines with the lowest Qpcr, the biomass detected by Qpcr. So we could see that the results of the 3 techniques correlated. On the other side, we can look at blade, where blade show high levels of infection. Blade didn't contain any gene for leaf rust. It only contained a stem rust resistant gene, but that's not effective against leaf rust. And with our Qpcr, you can also see high levels of the fungal biomass detected in these plants. So what we did is we looked at some of our breeding lines as well, where we looked at both the seedling stage and the adult plant stage. So remember, the seedling stage are being provided by the plant by the LR19 gene. So all the plants that contained the LR19 gene showed low levels of resistance. The lines that contained LR34 showed infection, but LR34 is not effective already at seedling stage. So this also correlated the data that we found here. And then if we look at the data for the adult plant stage, this time we want to look at the expression of the LR34 gene. And here you can see that lines that contained the LR34 also showed low levels of infection at adult plant stage, although some of them that contain it didn't show that lower level, but these low levels are due to the LR19 being present, and the LR19 helps the LR34 to provide resistance. So once again, our data correlated well. We did the same principle before the Fusarium head blight, where we looked at spray inoculation, and we looked at the beta-tubulin and the reference gene expression. We also looked at the expression of the Tri5 gene, that is for the mycotoxins that's produced by the Fusarium. And then we also compared that with our Fusarium head blight infection percentages. Unfortunately, the expression of the mycotoxin gene was not high, as you can see with the red line there, not a high level of expressions, but we saw a very good correlation between the Qpcr data and the infection that we had in the greenhouse. And the same trend was seen when we did the point inoculation. Then we can't do all of this without looking at the bread making quality. So we also identified the best lines with the highest number of resistant genes, and these lines we then screened for markers that's linked to the bread baking quality. And we screened our rust lines, and in our rust lines we found genes that has good bread baking quality and also dough strength, similar for our Fusarium head blight lines, except that there was a gene present that can have an effect on dough strength, so we would ideally like to eliminate that from the lines that we have. After we've done all this, the proof of the pudding is taking these lines to the field. So we did some field trials, and I had to get a nice picture from the internet. We don't have these nice field trials, but we did take it to the field. So we screened the original lines that we had. We used as parents, and then we selected lines with different combinations of the disease-resistant genes to see if the ones that contain more genes show higher levels of resistance within the field. So what did we see from our field trials, and I'm only going to speak about stem rust here, is that there was a very good correlation between the data that we had in the lab and the data that we had in the field. And that we saw that if we didn't detect any gene in the lab, those plants were very infected. They were moderately to severely infected by the disease. And then lines that we had more than one gene showed very high levels of resistance in the field. So once again, our marker data correlated well with what we've seen in the field. So what have we achieved thus far? We were successful in development of pre-breeding lines that contains resistance to both of the 3 rust and the Fusarium Haploid. We could implement the marker-assisted selection in selecting the best lines. We were able to incorporate the Qpcr technique, and we could see that we get a good correlation with the data using the Qpcr, and also the double haploid technique helps us to accelerate our breeding program. And we were able to develop a number of lines that are valuable with a number of genes, and some of these lines have already been made available to breeding companies to use in their breeding programs and to incorporate that into the cultivars that they release. So with marker-assisted selection, at this stage we have identified a number of lines that contains the rust lines with a high number of rust-resistant genes. We identified 2 lines that contained high number of Fusarium Haploid resistance, and now these lines were also tested, and they all have good breed-making quality. So we can use these now in our future breeding programs. So what still needs to be done? We need to take these lines that we identified and now incorporate some of the genes we lost throughout the process. You'll remember SR26, the source was not that good, but we did lose the LR19 as well. So we want to put that back into these lines. But also we want to add some additional rust-resistant genes, especially the stripe rust that we didn't have before. So we want to include that into the lines as well. We want to evaluate these lines for agronomic traits to see how they do in the field. Do they still have high yield? And can it be planted by the farmers? And we also want to, once we have enough seed, to do some grain milling tests and bread baking quality tests to confirm that these lines actually do have good bread making qualities. So throughout this whole process, it wouldn't have been possible. So I would like to acknowledge the National Research Foundation and the Winter Cereal Trust and the University of the Free State for providing funds to make this all possible. And then also some of the websites that are used for the pictures, especially FreePic. I don't have to do anything, I can just use those. So thank you for the websites as well. But all of this will not have been possible without all the students who worked on these projects. Some of them did both a master's and a PhD, some only a master's, some even did a PhD, a postdoc, and is now a lecturer here and is continuing with this breeding program. Thank you, Ansari. And then also some of the honors students and some of our current students. So the question, however, remains as to whether the pathogen or the wheat plant is currently winning this battle. And you can decide after my lecture whether it's the pathogen or the wheat. Thank you very much. Dankie, kia loboa.
Thank you, Liezel. Good evening, ladies and gentlemen. And now everybody expects me to say something insightful after all of that. And may you just keep in mind that Liezel was my mentor, so I should act smart and pretend that I know about everything that she was speaking about. So as Dr. Molapa said previously, that an inaugural lecture represents a significant milestone in the career of an academic, where a professor, in this case, Prof Herselman, is acknowledged, and she's also given the opportunity to share her groundbreaking research, innovative ideas, and deep insights into the world of agricultural serial killers. She shared this with the academic as well as the non-academic community. Apparently, Liezel, these lectures are not easy because of the high expectations, but congratulations, you made this look like child's play. Your profound knowledge on serial killers and how to stop them put you right at the top of the FBI's distinguished agents list. As was mentioned, South Africa is a net importer of wheat because wheat production is restrained by these difficult and destructive diseases that contributes to yield loss, grain quality reduction, and that incurs economic losses. Prof Herselman's research can thus be defined as a beacon of hope and relief because the contribution will provide an incentive to producers to plant more wheat, therefore improving wheat production in South Africa. Prof Herselman, this is a plea. Please do not hang up your racing shoes anytime soon. Keep on with the race against these destructive serial killers. The world's food security rely on researchers like yourself. Thank you very much. I just want to present. I was just asked to thank a few people. Firstly, Mr. and Mrs. Herselman, thank you very much for sharing Liezel with us. It's really a privilege, especially for me, to be able to stand here and talk about my mentor. So then, also thank you very much to everybody that took the time to attend, especially those that travel from far and the ones attending online. A special thank you to Dr. Molapo, I'm still trying to pronounce your surname, Kobela, for welcoming Prof Herselman and everybody, and also Prof Dani for introducing Liezel to those that don't know her, Alicia Pinar for arranging the event, and also for the technical support provided by Gavin Qpcr, Star Sound and Roystool. For the photographs, Devonish Photography is thanked, and also Pimento Montello for the catering of the food, and then of course Lumka for providing support from the department side. I think Lumka next week will be much relaxed after this. So without further ado, I would really like to invite you for some tea and refreshments in the foyer. But I would also like you to remain seated until Prof Herselman and her parents take their place at the door to be greeting everybody. So thank you and enjoy the rest of the evening.